Integral part of human circulatory system. Hold on. Yeah. Okay. So when we talk about it, it serves us with several functions, and very basic thing that if we have to point it out, like how the circulatory system supports us, correct? So we can come up with few of the examples. For example, circulatory system supplies blood, and the blood supplies oxygen. It supplies nutrition, correct? And it also removes the toxic waste like carbon dioxide or ammonia waste, etc. And it delivers it to our excretory system for filtration. So apart from that, what else does the blood do? The blood also functions as a very excellent defense mechanism, which contains a lot of immune cells and immune factors which help in protecting our entire physiology. Second thing, if we observe how the immune system, how the components of the blood support, if we have an injury and a small injury or a cut, it also helps in stitching it back so that the loss of blood is prevented in the form of coagulation or clotting of blood, right? And many other functions, like it also provides a very beautiful medium for mineral transport, or you can also movement of ions, movement of particles, and also the the destination where the hormones or the enzymes have to reach. It is an excellent medium for all these biochemical reactions to take place. Now, when we talk about blood, the components of blood becomes a very important factor for the whole of circulatory physiology, I can tell. Now, to understand this, first we will take the some amount of blood. Let's say that we take around 10 ml of blood, and we just subject this blood for the centrifugation process. Okay, so we centrifuge this blood, and after we centrifuge the blood, what do we get? We can see that the blood separates into three important layers. One is the lowest layer, and then above that you have one cloudy layer, and then you have the another layer which is straw color or yellow color liquid like layer. So these three layers, we can just give them names based on the composition. One of the lowest layer, the layer which sits at the bottom of the test tube or the centrifugal tube, it is called as red blood cells or the hematocrit layer. So this is where you have up to 40 to 52% of hematocrit in male, 37 to 47% of hematocrit. When I say hematocrit, that means red blood cells, students. Okay, so the red blood cells make up to 42 to 52% of the blood, and 37 to 47% of the blood in male and female respectively. Now, just above the hematocrit layer, what do you get is a cloudy layer. Okay, so which we call it as a buffy coat layer. So the buffy coat layer actually contains white blood cells and platelets. Whereas coming to the plasma is another important factor in our blood, where it contains water, proteins, nutrients, hormones, etc. So we'll discuss about what is a real composition in the coming slide. So this is the healthy blood. This is what the healthy blood looks like, and we understood that blood is not just made up of a fluid and then red blood cells. It contains white blood cells, it contains platelets, it contains red blood cells, it contains proteins, it contains hormones and enzymes, etc. Correct? So now coming to the detailed breakdown of what is the composition of the blood. First we talk about the plasma. So out of 100% of the blood, what do we have? We have around 46 to 63% of the blood which is made up of plasma. So what is plasma? You can see here it's a straw color fluid, okay, that is present in our blood. So what do this plasma has? Plasma is made up of 92% water. So majority of the plasma is water. So you can see that it is a very important fluid that helps, as a transport medium of all the biochemical ingredients that is necessary for our body. Now, the next component is the plasma protein. So the next major component in the plasma is the plasma proteins, which is made up of 7% of the plasma, right? So what are plasma proteins? Plasma proteins can be found as albumins, globulins, and fibrinogen. So if anybody asks what are these albumins, what are globulins, and what are fibrinogen, you should tell that they are a part of plasma proteins, okay? And they have a specific role to play. For example, if I talk about a plasma protein called as albumin, what do albumin do? They are very important for maintaining osmotic concentration and transportation of lipid molecules. Got it? Similarly, like globulins comes. In globulins, Again, we have three different types of globulins: alpha globulins, beta globulins, and then we have gamma globulins. Globulins are very important proteins when it comes to understanding the components of blood. Why? Because your textbook mentions about globulins as an immune factor. Okay, so a question can be expected on plasma proteins such as globulin. Okay, so we give a little bit more concentration on globulins. So if you talk about globulins, what do alpha globulins do? They maintain osmotic concentration and they also help in transport. When it comes to beta globulins, what do? They also transport and they maintain osmotic concentration. So which globulin is very, very important for us? Gamma globulins. So gamma globulins are also called as immunoglobulins. So this is where it's very important, where you write it down, right? So under plasma protein, we have albumins, globulins, and fibrinogens. Albumins, they maintain osmotic concentration and they transport lipids. And then when it comes to globulins, we have three types of globulins: alpha, beta, and gamma. Alpha and beta, they maintain osmotic concentration and they help for transportation, whereas gamma globulins are also called as immunoglobulins. Okay, and they are formed because of plasma cells. It's fine, but you remember that the gamma globulins act as immunoglobulins. What is immunoglobulins? They help in defense mechanism. Okay, these proteins are responsible for defense mechanism. Very important, students, so you don't forget this. Plasma, globulins, in that gamma globulins, and the gamma globulins being immune responses, okay, which help in immune response. Are you able to follow, students? Yes? No? Is it fast or should I slow down? You can give a thumbs up if you're okay, or thumbs down if you want me to slow down. Yeah, perfect. Okay, so I will go further. Okay, if you have doubts, please make sure that you put it in the chat box. I'll observe it, okay, during the class. So now we understood. Okay, this is very, very important. So I expect you guys to note this down. And the third type of protein, what do you have, is a fibrinogen. What do fibrinogen do? It helps in something called as hemostasis. Hemostasis is also called as blood clotting. You know, clotting of blood, that is stopping of the blood from leaking. So that is done by fibrinogens. So fibrinogens, gamma globulins, and the albumins, all of them belong to the class of proteins called as plasma proteins. Clear? Chalo. And we have regulatory proteins such as hormones and enzymes. And then we have other solutes such as nutrients, gases, dissolved gases, and water. Okay. So these are all general. So it's okay that we don't remember this much, but cool. Next one we go, that is the formed elements. What are formed elements? Formed elements are the cellular part of your cell. So this is also an important term that is necessary for you. What is blood made up of? Blood is made up of plasma and formed elements. Correct, no people? So if you just look at this, what do we have? We have plasma here, right? And then we have the rest of the two regions that are made up of cells. So these cells are called as formed elements, okay, which are produced in the blood on different parts of your body. Now, what do formed elements have? Obviously, we call it as red blood cells. Correct, right? Formed elements can be red blood cells, white blood cells, and then we have platelets. Correct, right? So first, let's discuss about this major part, that is the red blood cells. The RBC, it is also called as the erythrocytes. So they make up to 99% of the formed elements. Oh, sorry. They make up to 99% of the formed elements. Okay. So where are they produced? They are produced in red bone marrow. Very important. Okay. And you know what is the use of erythrocytes or the red blood cells? They are very important for the primary transport cells of oxygen and some carbon dioxide. I can't tell complete carbon dioxide, but it is some letters are not visible clearly. I can understand, but I will share this PPT with you guys, a link with you guys. You can download it from the Google Drive. Okay, so no problem. You have to magnify this because the information is a little large. Okay, but I'm telling you, if you don't understand my speech, for writing it down as specific ones, I can just point it out. Okay, I will just write it down again. Chalo. So when it comes to red blood cells, what do? They transport O2, correct? And also CO2. I can't tell that CO2 is a major part because we have seen with respect to breathing and exchange of gases, how much of carbon dioxide is carried by red blood cells, correct? Next one, leukocytes. What are leukocytes? Leukocytes are also called as WBCs generally. So in leukocytes, what do we have? We have granular leukocytes and we have agranular leukocytes. The names, the description, and how they participate in an immune response, we are going to study in the further slides. So, JNV Ramnagar students who are worrying about not visible, no, not being visible, don't worry. Okay, next slides are coming up. And next one, what do we have? We have platelets. And platelets, the role of platelets also we'll discuss again. So you understood, right? What we have to remember from this slide? Plasma and then formed elements. Plasma, in that we have to give more importance to albumins and globulins. And then in globulins, I told you about gamma globulins, which are acting as immune response. Done? So formed elements, what are formed elements? They're made up of erythrocytes, leukocytes, and then platelets. Done. Now let's go for the next slide. See students, when we look at the way in which these cells are produced, it is very fantastic to understand. They all originate from a single cell. That is when we understand this meaning of totipotency. What do we tell totipotency? Totipotency means you have one cell, okay, one mother cell. We'll understand here now. We have one mother cell. As it divides and divides, the daughter cells, they specialize to become some important cell. So this one cell which forms into different elements, okay, for example, it can become red blood cell, it can become white blood cells, or it can become antibodies, anything. You see, the varieties are very, very high, what they can become. So this capacity of a cell to become any functional cell is called as totipotency. So one mother cell, one major cell, where all the formed elements, whatever we have seen here, that is the erythrocytes and the leukocytes and the platelets, all of them originate from a single cell. Okay, so we are talking about how formed elements are formed. Okay, now we can, if I ask you the question, okay, how are red blood cells formed? What do you tell? You tell that red blood cells are formed in bone marrow. Who told the bone marrow to produce red blood cells? It can produce anything. The same bone marrow, red bone marrow, produces some of the elements. Now that will become a tricky part, isn't it? That is why we have to understand how the formation of these cells takes place. Okay, so let's understand this. So formed elements, all of them start with single cell, which we call it as multipotent hematopoietic stem cells. What is hematopoiesis? Hematopoiesis or hemopoiesis, what we tell, it is the process of formation of blood cells. Since we are talking about hematopoietic cells, they're like different blood cells are formed. Okay, so that is the meaning of that cell. So what do we have? The first cell, the first cell is called as multipotent hematopoietic stem cells. Okay, and this multipotent hematopoietic stem cells are also called as hemocytoblast. I think you can read this right, hemocytoblast. So this is the mother cell. Now, what does hemocytoblast or multipotent hematopoietic stem cells do? They divide, okay. So when they divide, now what happens? One of the daughter cell will specialize into myeloid stem cells and another daughter cell will divide into lymphoid stem cells. So these are major stem cells. You know what are stem cells, right? They are totipotent cells. So which are the two major stem cells here? Myeloid stem cells, lymphoid stem cells. So this is where your part of remembering starts. You have to remember from down here. Okay. Now listen here. We'll talk about the myeloid stem cells part, and then we'll talk about the lymphoid stem cells part in a little while. Okay. First, myeloid stem cells part. What do myeloid stem cells do? They divide, right? Again, the myeloid stem cells divide. Let's say it divides into 4 different daughter cells. So each of the daughter cells will again specialize into parent cells. What are those? Myeloid stem cells will specialize into megakaryoblast, proerythroblast, myeloblast, monoblast. Okay, let's go again. We have multipotent hematopoietic stem cells. It divides into 2 important cells. What are those? One is myeloid stem cells, other one is lymphoid stem cells. When it comes to myeloid stem cells, we have 4 varieties. What are those? Megakaryocytes, proerythrocytes, myeloblasts, and monoblasts. It is very easy to remember if you go to the what is their product. Okay, so let's see how each of the cells and what they give us. Now, if I talk about megakaryoblast, what does it do? It produces, blast will become site. Okay, megakaryoblast will give rise to megakaryocyte. Megakaryocyte will fragment into platelets. So if I ask you what is the parent cell of platelets, what do you tell people? It is megakaryoblast. And how are the platelets formed from? It is formed through megakaryocyte. Did you understand? Where do we get platelets from? Megakaryoblast. What group of stem cells they belong to? Myeloid stem cells. Okay, got it? Now, when we talk about the next one, that is proerythroblast. Pro is the starting, okay? Erythro means red blood cells. Blast means, mother cell. Proerythroblast will give you what? Here, site is there, but look at the name. It is called as reticulocyte, students. Okay, I will erase this so that you can copy it down. Why reticulocyte is very important for us? Reticulocyte is also called as immature RBC, and it has nucleus. Okay, it is around 0 to 40 days old. Okay, it will live for 0 to 40 days. Okay, so proerythroblast will give us reticulocyte. Reticulocyte are called as immature RBCs. They have nucleus. Got it? So it will later mature after 40 days. What it will become? It will become a functional erythrocyte or red blood cell. Okay, so done. Like, I hope you have understood till this part. I'll go for the next one. One is myeloblast. So megakaryoblast done, proerythroblast done. Next is myeloblast. Myeloblast is very easy. Myeloblast will divide into three types of white blood cells. What are those? Basophils, neutrophils, eosinophils. Okay. So finally, we have monoblast. What do monoblast give us? Monocytes. Each of these basophils, neutrophils, eosinophils, and monocytes, their functions we'll talk in a while, students. Okay. Is it clear who originates from where and what are their mother cells? Yes, no? Are you following, students? Students, did you understand? All right. Okay. Virendra is fine. Okay. You just let me know. Okay. So I will continue. So now if you look at the other part, what is our other part? Our other part is the lymphoid stem cells. Myeloid is done. So if you just ask you, what does myeloid stem cells give? You have to tell four cells. Remember this, correct, right? Megakaryoblast, proerythroblast, myeloblast, monoblast. Very simple. Now you know what are the cells are coming from where. Now coming to lymphoid stem cells, what does it give to? You know that lymphoid stem cells means it has to give us blast, so it will give us lymphoblast. Now, lymphoblast will divide into again two types of cells. One is NKC cells, we call it as, or natural killer cells. And then we have small lymphocyte. Okay, it is also called as small lymphocyte cells, which finally divides into T lymphocytes and B lymphocytes. So the function of these cells will be discussing later in a while, next slide. But I hope you understood how these formed elements are formed. Okay, I can go to the next slide, I believe. Let's go. Now, I asked you a question. Who tells this cells? Who tells this mother cells? Who are those multipotent stem cells? Who tells this multipotent stem cells to exactly become either lymphoid stem cells or myeloid stem cells? Okay. Or who tells these myeloid stem cells to become whether proerythroblast or to become monoblast or myeloblast or megakaryocytes? Who tells them? Correct, right? There has to be a feedback. There has to be a mechanism who is guiding the cells to give us exact amount of cells. We don't see our blood overflooding with red blood cells in a normal condition, or we don't see our cells overflooding with platelets. There are specific numbers and specific ratio of these blood cells in our body, right? So that is why there is a regulation mechanism on how much amount of blood cells have to be produced. Okay, so who does that? So we call them as hematopoietic growth factors. So hematopoietic growth factors are such factors which promote the growth of certain blood components or blood factors. Example, we have erythropoietin, which is produced in the anterior part of your kidney. Okay, erythropoietin, what it will do? It is also called as EPO. Erythropoietin, it stimulates the formation of red blood cells. Okay, now we have thrombo. Okay, 1 second. I think we have a question. Yes, JNV Ramnagar, please tell me. I think you have a message for me. Let me see. Sir, letters are not visible. Once again, previous slide. All right. So previous slide is where the, mm, formation of the red blood cells took place, right? Okay. Let me see. You want me to explain this? Hold on, let me erase all the ink. Mm, yeah, now it's clear. You hold. I'll just listen to you. 1 second. What's your doubt? Yeah, JNV Ramnagar. You want me to explain the slide again? If it is so, give it a thumbs up there, or you just let me know what's your query. Yes. Okay. We'll go again. No problem. Huh? So, see, students, I was talking about how the cells are formed. Okay, it starts all the way from one cell. What is that one cell? It is called as hematopoietic stem cells. I told you what is hematopoiesis. Hematopoiesis is a process of formation of blood cells. So that means, how do you remember hematopoietic stem cells? That's it. And one name we have given multipotent. Why multipotent? Because it gives rise to so many different types of cells. Okay, simple. So it starts from hematopoietic stem cells and it gives rise to two important stem cells. What are those two classes? One is myeloid stem cells, other one is lymphoid stem cells. Now, coming to myeloid stem cells, what does it give rise to? It gives rise to four different types of daughter cells. These are also mother cells. Okay, what are they give rise to? Myeloid stem cells will give rise to megakaryoblast, proerythroblast, myeloblast, and monoblast. Okay, come again. Megakaryoblast, proerythroblasts, myeloblasts, monoblast. Very simple. Megakaryoblast will give rise to megakaryocyte and it will give rise to platelets. Okay, megakaryo. Whenever it comes to mega, you go for megakaryocyte and it finally gives rise to platelets. As simple as that. Whenever you come across a word called as proerythro or erythro, that means red blood cells. Proerythroblast will give rise to what? Reticulocyte. What are reticulocytes? They are called as immature RBC. Correct? No? Immature RBCs. So why do you call it as immature RBCs? It still has less cytoplasm and nucleus. Okay. So it takes 40 days. Okay. Around 40 days, 0 to 40 days for your reticulocyte. There was a question, what is the name of immature red blood cells called? Or what is the name of nucleated red blood cells called? Okay, answer was reticulocyte. Okay, reticulocytes are nucleated and they are very immature. They don't do the function of carrying oxygen. Okay, so they lose all this nucleus and eventually become active for carrying our oxygen. Okay, so these are called as reticulocytes, and reticulocytes will further mature into erythrocyte. You know erythrocyte, they are called as red blood cells. Now coming to myeloblast give rise to immune cells. So immune cells, your WBCs can be divided into basophils, neutrophils, eosinophils, etc. Okay. So when you talk about myeloblast, they give rise to three types of immune cells based on their behavior with the we'll talk about it. Okay, so it gives rise to basophils, neutrophils, and eosinophils. Finally, we have monoblasts, which give rise to monocytes, which are again immune cells. Okay, this is the part of myeloid stem cells. So if I ask you what does myeloid stem cells give rise to, it is platelets, red blood cells, basophils, eosinophils, neutrophils, and then finally monocytes. Done. And next we go for lymphoid stem cells. What do lymphoid stem cells give rise to? Lymphoblasts. Lymphoblasts will give rise to two different type of cells. Natural killer cells, very important this one. Lymphocytes are the parent cells for natural killer cells. Everybody forgets this part. Everybody thinks that it is coming from myeloid stem cells. No. When you talk about lymphoid stem cells, you have to give credit for the lymphoid stem cells for forming natural killer cells. Okay. And finally, when it comes to other cells, that is small lymphocytes, which will eventually divide into two important cells, T lymphocytes, B lymphocytes. Got it? So if I ask you what do we get from lymphoid stem cells, what do you tell? You tell natural killer cells, T lymphocyte cells, B lymphocyte, T, B, and NKC. Done. So this is about the slide that you asked for. I hope you understood. Is it clear? Or should I repeat again? Yeah, perfect. Chalo. Next, I was talking about, Ramnagar student, you put your hand down, okay? So that next time when you again put it up, I can understand that you are asking me something. Yeah, no problem, okay? Just ask me. We can understand clearly. That's important. Okay, so now I told you how the factors are formed, right? We were explaining a story, like who tells who to, how do they produce. So when we talk about factors, they're like proteins, okay, signaling proteins. They go and tell the stem cells to divide and differentiate. Okay, dividing means how they become, and differentiate means becoming specialized. So similarly, we have erythropoietin as the first factor. So this erythropoietin is produced in the kidneys, and they help in the formation of red blood cells. When you talk about thrombopoietin, they help in the formation of platelet. OPLATLAT. Oh, I don't know. Okay, so I'm not getting the spelling. One minute. Spelling one nimsha. Huh? So platelets. Next we have cytokinins. Cytokinins very important again. Okay, so they are responsible for immune reactions. Okay, for example, if they have to form WBCs, like colony stimulating factors and interleukins are two important factors. They are responsible for immune cells. Okay, immune cells. Remember this interleukins, name of this interleukins and colony stimulating factors, and this name called as cytokinins. What do cytokinins do? They help in the formation of immune cells. So three names are important here, not the colony stimulating factors and interleukins. Very, very important are three. What are those? Erythropoietin, where is it formed? Kidney. What do they do? Red blood cells formation. Thrombopoietin, what do they do? They stimulate the formation of platelets. What about cytokinins? Cytokinins have two more factors. One is colony stimulating factors. Next one is interleukins. So overall, what do cytokinins do? They help in the formation of immune cells. What are immune cells? Basophils, eosinophils, neutrophils, okay, monocytes, natural killer cells, etc. Got it? Yeah. So these are called as hematopoietic growth factors. These three names, you take it down. Okay. I hope you have taken it down, and I'll move to the next part. This chart, I know it's not visible. You can get a very quick look at it and understand the role, position, what do they do, and how many numbers, everything. It's in one chart. As I told you, I'll give you the PPT. You just look at this information and go through before examination. Okay. Now, coming to the detailed observation or understanding how these cells function, we have to start from red blood cells again. So, if I talk about red blood cells, right? So red blood cells are very, very special, right? Why? Because they are biconcave in shape, right? And very small cells, and they're enucleated. Enucleated means what? First they had nucleus, and now they don't have nucleus. They are enucleated cells, and they have a special plasma membrane. Okay, they have a special plasma membrane. So what is this special plasma membrane? Have you ever wondered, students, when the blood vessels are there, as they move towards away from the heart, they become smaller and smaller and smaller. So the larger arteries becomes smaller arteries branches to become arterioles will again branch to become capillaries, capillaries to microcapillaries. So they become smaller, extremely small. So at one point, it becomes so small that your red blood cells, even though they are around 100 micrometer in length, they have to really squeeze through this. They have to squeeze through these blood vessels. As when they squeeze, what happens? Their shape of this biconcave shape, like coins, they can get folded. They can get folded or squeezed. What happens when you just squeeze something, it gets folded. So that folding happens, okay. But when they are coming out of that place, they regain their shape back. And the reason is because of the nature of the plasma membrane that the red blood cells have. They have a specialized elastin protein that gives them the flexibility to bend and regain its shape. Okay. Now, coming to the red blood cell itself, what is present within the plasma membrane is most of the protoplasm is filled with this metalloprotein. We call it as, what do we call it as? Metalloprotein. Now, who is our metalloprotein? You guessed it right. It is hemoglobin. Now, what the structure we are looking at is the hemoglobin. So, this metalloprotein becomes a very functional unit of our red blood cells. So, if I take hemoglobin, one hemoglobin, and then if I look at it, what do I observe? I see that my hemoglobin is a globular protein. What is globular? It is the structure. Okay, it is the structure. So globular, why do we tell globular protein? Because of the complexity of the structure. Why do we say it is complex? Because proteins are made up of what? You see, what are proteins? Can I ask you this question? What are protein made up of? Let me see. If you have answer for this, then I will understand. What are proteins made up of, students? What are they made up of? What are proteins made up of? We studied biomolecules, didn't we? Yes. Amino acids. Okay. I have one more question. Now, the sequence of amino acids forms what? Amino acids are the monomers. So now I have a chain of amino acids. What do I call them? What are amino acids made up of? I mean, like, what do they make up to? You have an answer? Let me wait. Yes. Tell me. I wait. No problem. Are Yeah, polypeptide chain. Exactly. This is the answer I was looking for. Okay. So they're made up of polypeptide chain. So if you see the nature of polypeptide chain, we have four polypeptide chains. Okay. In a hemoglobin. What do we have? We have identical chains. That means they look alike. Alpha chain, see, non-identical alpha alpha. See, alpha chain, beta chain. So these four polypeptide chains, they come together and form a specific structure, which we call it as a globular structure of hemoglobin. Now, if you look at this, these polypeptide chains, they have this cofactor, which you call it as a heme group. So this heme group as an ion or a ferrous or metal ion, so which helps in carrying of oxygen. So each hemoglobin can carry up to 4 molecules of oxygen. So that is the structure of hemoglobin, and that is what makes the RBC functional. Okay, so this is about RBC. Now we have a map of where the RBC is formed. Now you can see this hematopoiesis of hematopoiesis of erythrocytes. When I tell, it always starts at hematopoietic bone marrow. When I say hematopoietic bone marrow, they are all called as long bones. Okay, long bone. Which is your long bone? The one that is present in your pelvic bone. Pelvic bone is a long bone, and then the bones of your thighs and the bones of your shoulder. So this is where the site of your formation of red blood cells take place, right? So we know that they are produced as reticulocytes, and further they mature into functional red blood cells. Functional red blood cells. Usually, what happens is when the red blood cells are formed, as I told you, they have a specific expiry date. So each of the red blood cells can survive up to only 100 and only 120 days of lifespan is there for a red blood cells. So what happens after red blood cells die? See, red blood cells, they have very important components in it, which we call it as the protein factor. Correct, right? This hemoglobin is a very, very important ingredient of the red blood cells. So we can't just waste the red blood cells. So that is why when the red blood cells come to the end of their life cycle after 120 days, they have to be recycled somewhere. So that is when we have a cyclic event that is happening where the old blood cells are sent for their death, and the remnants of the red blood cells are used for the construction of new red blood cells. Are you understood, right? We take the old parts, and then we discard whatever it is old, and we put it in the new. So that is what is happening in the formation of red blood cells. So you see, it starts producing in the long bones. So the long bones are also called as hematopoietic bones. So here what happens? The stem cells will differentiate into erythroblast. Erythroblast will give rise to reticulocytes, which are immature red blood cells. And finally, they give rise to mature red blood cells. And we know that after 120 days, the red blood cells will become old, and they are transported to spleen. Remember, where are they transported to? Spleen. So I think you would have heard about what is the other name for spleen? Spleen is also called as graveyard. Okay, graveyard of RBC. So it is where the red blood cells go and die. So how do they die? Now we have some of the cells which act like amoebas. They go and eat the red blood cells. And when they eat the red blood cells, what they do is they break the red blood cells in such a way the hemoglobin of the red blood cells are separated from the cell factor. All the cells get digested, only the hemoglobin remains. Understood? Now, this hemoglobin is again sent back to your liver. Okay. So sometimes what happens, this heme is too much in your body and your body don't want it. So that time what happens, if you see your fecal matter, okay, the waste, what we excrete, you see that it's brown color. So the brown color is because of this heme group that is excreted out of your body in the form of bilirubin and biliverdin. Are you following me, students? Okay. And sometimes what happens, the same heme group, the iron ions which are extracted from it, will be transported back to your long bones so that the reproduction or reuse of this iron ions happens for the construction of new red blood cells. I know that this is too much to understand. All you have to remember according to your examination is where are the red blood cells are produced. They are produced in the long bones. Done. What are the long bones? Your shoulder bones, your thigh bones, your pelvic region, pelvic bones. So these are called as long bones. How are they produced? They are produced by the stem cells, erythroblasts, and then it gives rise to immature cells called as reticulocytes, and then finally it matures into red blood cells. What is the lifespan of red blood cells? 120 days. Done. Now, of what happens after 120 days? It goes for the spleen for degeneration. Okay. So this is the very important things that you have to remember. Okay. Now, another important thing is once the heme group, what happens to the heme group? Heme groups are sent to the liver where it is sent as biliverdin and bilirubin. Okay, so the liver takes it and either it decides to excrete or it decides to give it for the production of new red blood cells according to your body's needs. So this is about what is happening with the life cycle of red blood cells. Is it clear? So that when you look at the chart, you will understand, you will start connecting it as a story, and you don't have to remember the facts like 120 days or the long bones or something like that. Correct? Did you understand this? Can we go for the next one? Yes or no, students? Are you able to understand? Yes. Okay. So let's go for the next slide. In our next slide, this is about the immune reaction. We'll talk about it. Okay. So usually what happens is whenever the bacteria, for example, I have a prick, okay? See here, there is an open wound, and then all this bacteria or the fungal spores or whatever, they have entered into your tissues. Now, these what happens is they release some of the factors, okay? So this some of the factors that are released by this microorganisms presence are recognized by your leukocytes, that is the white blood cells. There are different types of white blood cells such as eosinophils, basophils, monocytes, neutrophils, etc. So what they do is they squeeze out of your blood capillaries, they attack these microorganisms and then they neutralize it. So this is not as simple as what I tell. There is a very elaborate mechanism for how these cells function. Okay. Anyways, we have to remember certain facts. Okay. So when you talk about leukocytes, we have three important ones. One is neutrophils, eosinophils, and basophils. So this is the exact microscopic image of how they look like under the microscope. You can see that there are lots of granules within the cytoplasm. That means they are called as granulocytes. And you have to also give a look at the shape of the nucleus or how many lobes the nucleus has. So these are some of the important morphological features of the cells that help us to identify what cell is what. Anyways, I know it's hard to gather any of this information. Let's understand about leukocytes. What are leukocytes, students? They're called as WBCs, correct? So I told you earlier that we're going to discuss very detailedly about it. Let's discuss it now. So when you talk about leukocytes, based on their appearance, you can see here, they are divided into two important types. What are those? One is called as granulocytes. Granulo means granular. So if you look at the picture, they look like small granules that are present within the cytoplasm. So they are called as granulocyte. And if they don't have large granules, see, even agranulocytes have granules, but it is very negligible size, very small size. That's why we call these cells as agranulocytes. the classification of granulocytes, what type of cells are there in granulocytes, what type of cells are there in agranulocytes are very, very, very important. Okay. So I hope you take the examples of it. Okay. So when you talk about granular leukocytes, as I told you, they have abundant granules, and the examples are neutrophils, eosinophils, and basophils. Remember this, okay? Neutrophils, eosinophils, basophils are the examples of granulocytes. Now, coming to agranulocytes, what are the examples? Monocytes, okay, which is or you can tell macrophages or lymphocytes. If they give you macrophages, lymphocytes as the two options, you have to go for this. Macrophages and lymphocytes are agranulocytes. Remember this. Why? Because the monocytes are the one which develop into macrophages. Again, monocytes are agranulocytes. So what are the examples for agranulocytes? Monocytes, macrophages, lymphocytes are the examples for agranulocytes. Okay. So now when we talk about where the granular leukocytes are produced, again they are produced in red bone marrow. Okay. And what is the nature of agranulocytes? Sorry, granular leukocytes or granulocytes? They have very short lifespan, from few hours to few days. That's all. And after that, their numbers are reduced when the infection is disappearing. Okay. Now, one of the key characteristics of granular leukocytes is when you observe it under the microscope, their nucleus is always lobed. You see, it's not like nice, round, and spherical. You can see that they have two lobes like this or multiple lobes like this. Okay, something like this. So that is the shape of the nucleus they have. So they contain lobed nucleus, and also based on how they react to the stain. For example, you have neutrophils for neutral strain, basophil for basic strain, eosinophil for acidic strain. So based on how they react to what stain that we use, we can classify them as basophils, neutrophils, and eosinophils. Clear? So can we go for the first one? What are neutrophils? Characteristics of neutrophils. Very important points are there. You have to remember this, please, students. So I hope that you just make a quick notes of it. Okay. Now, coming to neutrophils, this is the first example of a granulocyte. So they are the highest or abundant granulocyte that is present in your granulocyte is which one? Neutrophils. 50 to 70% of the granulocytes are made up of what? Neutrophils. So they react to the neutral stain. So that is why they are called as neutrophils. Okay. Now, what is the nature? When you look at the neutrophils, 2 to 5 lobes are present within the nucleus. So in the nucleus shows, see, there are 2 to 5 different lobes that are present within the nucleus. That is why we call it as lobed nucleus. Okay. Now look here. So if you look at the nucleus, they are lobed. How many lobes are there? 2 to 5. Remember this, 2 to 5 lobes. Which cell it is? You go for neutrophils. What type of neutrophil it is? It is granular leukocyte. How much percentage? 50 to 70%. Highest number. Okay. So the numbers increase when as the mature. Okay. So since the nucleus shows a different character of having 2 to 5 lobes, they are also called as polymorphonuclear cells, PMNC. So they usually the exams ask with these terms. The PMNC has how many lobes? What is the name of PMNC? Neutrophils. Why it is called as PMNC? Because it has around 2 to 5 lobes. Poly means what? Sorry, what is poly means? Many. Morpho, appearance. Nuclear, okay, or nucleus, that is nucleus, already. So C means cells. Polymorphonuclear cells. So these are called as what? Polymorphonuclear cells, are also called as neutrophils. Now, when do you see neutrophils? Whenever there is a bacterial infection. Remember this. Okay. Whenever they have a bacterial infection, their numbers increase. So what do they do is, when they sense that there is a presence of bacteria, so these granules that we talked about, so this is the cell. We saw that there are small granules that are present within the cytoplasm. Inside these granules, what do you have is the lysozymic enzymes. You know what are lysozymes, right? They are hydrolytic enzyme, or which is also called as suicide pouches. So they contain highly acidic digestive enzymes which kill or degenerate the bacterial cells. So they suicide themselves. They just go attack this bacteria and release these lysosomes as they consume them. So the cell gets digested along with these neutrophils. Okay. So when are the high? So when can you see neutrophils number very, very high? Whenever there is a bacterial infection. Very, very important. Okay. Bacterial infection is there, you see a lot of neutrophils. So sometimes because of drug toxicity, the neutrophils numbers can decrease. So this is about neutrophils. Is it clear? Okay, this information is very, very important. Are you following? Virendra and JNV Ramnaga? Yes? Yes. Good. Thank you for the response. So let's go for the next one. Thank you, Virendra. Yeah. Sure. Coming to eosinophils. You see, eosinophils are very easy to remember because eosin, you might have used this stain. Eosin is a stain. And it is what stain? It is an acidic stain. Oh, it's already given here. See here, they are only 2 to 4% in the overall granulocytes, and they are acidic stain, and they are called as eosin. And if you see the lobe nucleus, the nucleus is only 2 to 3 lobes. Okay. And the key characteristics of eosinophils, you see here, they are antihistamine in nature. What is antihistamine? See, histamines are allergens. Okay. What are histamines? They are allergens. So whenever your body comes across a parasite, or whenever you have pollen grains or the dust, you keep on sneezing because of the allergic reaction, or you start itching everywhere, or it's burning or rashes because you had some gobi manchuri in the street, or you have a food poisoning. All those are allergy reactions. So allergy is caused because of the release of histamines. Okay, so these histamines, what they happen is they are increasing this immune reaction. Now what happens is eosinophils, they have antihistamine in them, so which neutralize this histamine activity, or reduce this allergic activity. Okay, so now they also regulate the function of basophils and mast cells. They also control how the basophils and mast cells also should act upon. So when their eosinophils are produced, they are produced during the parasitic worms. Very, very important. Whenever you have parasites like worms in the stomach or worms in the liver, so at that point you can see that the production of eosinophils is very, very high. Okay, and they're very, very specific. So they attack the specific components, and then they destroy it. So they're highly specific. Now, when can you see high counts of eosinophils? Whenever there is an allergy, whenever there is a parasite infection, or autoimmunity. Autoimmunity means self-destruction, students. Okay. Okay, what is self destruction? Your cells, your own body cells, think that your cells, other cells, are enemies, so they start attacking your own cells. So that is called as autoimmunity. That means failure of immune system to recognize which is my cells and which is outside cells. Okay, so at these three, allergy, parasites, and autoimmunity, you can see that the eosinophils numbers are very, very. Got it? Eosinophils, they react to acid strain, that is eosin. The nucleus is 2 to 3 lobed, very, very important. They are antihistamine in nature. So basically, they control basophils and mast cells. And what is the other important thing? When are they produced? Whenever there is parasitic worms, whenever there is allergy, whenever there is autoimmunity, you can count on eosinophils number very, very high. Now, the final one, basophils. When it comes to basophils, they are less common, very less in number, 1%, very small compared to that of neutrophils and eosinophils. So, since basophil, basic, that's why they react to the alkaline stains. Now, these basophils, they are having very large granules, usually dark blue stains. Okay. Now, they contain up to only fixed number of lobes. There are only 2 lobes are present in the nucleus. Now, when are these basophils produced? Inflammation. What is inflammation? Inflammation means swelling. Whenever you have injury, whenever you have a cut, whenever your tissue has been damaged, this is called as what? Inflammation reactions. So at that point, you can see a lot of basophils reacting. Okay. Now, what do they do? Granules from the basophils, they release histamines and heparin. What is histamines? the allergic reactions, they tell, oh, something is attacked, you should just come and neutralize all this bacteria, there has been a cut. Those are called as histamines, you see. So when the basophils release histamine, that is when your eosinophils come, they're antihistamine, right? So when it is not necessary, eosinophils control basophils, so they're histamines. And heparin, what is heparin? It is anticoagulant. Anti Anticoagulant means what? Anti means no. Coagulation means what? Clotting. So they prevent clotting of blood. So heparin and histamines are released by who? Basophils. Now, when can you see more of basophils? Again, same thing. Whenever there is allergy, whenever there is parasitic inflammation, or sorry, parasitic infection, or hypothyroidism, very, very important. Hypo means what? Less. Thyroidism what means what? Your thyroid glands, they release some hormones. So whenever they are suppressed, you can see the increased number of basophils. So usually the low counts of basophils can be seen in pregnancy and whenever there is stress or there is hypothyroidism. So this is about basophils. Did you guys understand today's session? Is it fine? Yes, students, or no?